If I asked you to name where light comes from, you would say things like the sun, a lamp, a screen. That is why what I'm about to say sounds almost impossible. The truth is that living organisms emit light. You, me, plants, animals, even cells in a dish, all releasing ultraweak photons, often called biophotons. Not a glow that you can see, it's something far fainter than that. Today, I want to go over a few common questions about biophotons and what recent research is starting to show. If biophotons are a type of light, why can't we see it? The thing about biophotons is that they're actually not entirely outside the visible range. In fact, it spreads across a broad band of wavelengths, often described as roughly 200 to 800 nanometers, reaching from the ultraviolet into the visible range. The answer has to do with its intensity. The photon intensity is far below what the human eye can detect. A common number you will see reported is on the order of 1 to 100 photons per second per square centimeter. I know, it sounds like a lot, but it's actually not enough to be registered by the human eye as an image. Under ideal conditions, humans can detect a tiny flash of light when only a handful of photons are absorbed by rod cells. But forming a stable picture of something, that's different. It takes far more photons spread across space and time. Biophoton emission is simply too faint and too sparse for that. To measure these photons, scientists have to be pretty crafty and particular. Essentially, they build a mini world where no light exists but your own. In order to measure someone's biophotons, they would not stand in just a normal dark room. They would go into a light, tight chamber built to keep stray photons out. Then one would wait through a dark adaptation period, often on the order of 10 to 20 minutes, basically given the body time in darkness so any lingering effects of recent light exposure can fade down. Only after that does the actual recording begin. And when it does, the tools are very specific. For photon counting, researchers often use a photomultiplier tube, a detector designed to register single photon events. For imaging, they use ultra-sensitive cooled cameras, like cooled CCD or EMCCD systems. That way they can map where emission is stronger or weaker across a surface. So no, the human eye cannot detect something this dim, but specialized instruments luckily can. That being said, other organisms may be able to because they have visual systems that operate near the edges of physics. Two cool examples of extreme low light specialists are tarsiers, which are built for nocturnal vision with rod heavy retinas and enormous light gathering eyes. And then there are silver spiny fins, a type of deep sea fish that has visual systems that are adapted to extract visual meaning from the thinnest possible trickle of photons. Now I want to turn to a deeper question. Why do organisms emit light in the first place? The first thing to know is that biophotons are not the same thing as bioluminescence. This is not a special glow system with a dedicated purpose, the way a firefly has one. In most mainstream explanations, ultraweak photon emission is treated as a byproduct of ordinary life chemistry. And because living systems are not calm, chemically speaking, they are full of reactions. Electrons moving, bonds forming and breaking, energy being transferred from one molecule to another. And in that constant activity, a small fraction of reactions briefly create molecules in excited states. That is the key phrase, excited state. It simply means a molecule has been pushed into a higher energy condition, and what goes up must come back down. And when it relaxes back, One way it can release that extra energy is by emitting a photon. So, where do all those excited states come from in biology? A major pathway researchers focus on involves oxidation chemistry. When oxygen is involved in metabolic reactions, it can generate high energy intermediates. It can also generate reactive oxygen species, which are a normal part of metabolism, especially in mitochondria. Those oxidative reactions can set off cascades, including lipid peroxidation, where membrane lipids undergo chain reactions. And in the middle of all that chemistry, you can form excited carbonyls and other excited products that can release photons as they return to a lower energy state. And that is why the literature often links ultraweak photon emission to oxidative metabolism, not as a single cause, but as a common route. Mitochondria are one of the major sites where oxygen is used, where electrons are passed along, and where reactive oxygen chemistry can be generated. So they are a plausible contributor for sure. But in general, it is rarely framed as one organelle doing all the glowing. A better way to think about it is distribution. Many reactions in many places, each contributing a tiny piece. And the broad spectrum we talked about earlier supports that feeling. If the emission spans a wide range of wavelengths, logic would say you are not looking at one single emitter. You are looking at multiple chemical pathways and multiple excited species, each leaving its own faint fingerprint. This is what made researchers ask the next question. If biophotons are tied to chemistry and metabolism, does its emission change when our state changes? One of the easiest ways to test this is to stress the system and to see if the emission responds. In skin studies, for example, researchers apply a controlled stressor that is known to shift oxidative chemistry. Then they measure photon counts or images before and after. What they tend to find is not a dramatic flare of light, but a measurable shift. The emission pattern changes with the stressor, and in many setups, it changes in specific regions. In imaging studies, that can look like certain areas brightening relative to their own baseline, creating a before and after map that is different enough to quantify. When the stressor is removed or time passes, the signal may settle toward baseline again. That matters because it is one of the simplest ways to connect the signal to biology. You push a known physiological process, and the emission moves with it. Another clean move is to look for rhythm. The body is not chemically constant across the day. Metabolism, hormones, temperature, circulation, it all shifts. So researchers measure at multiple time points and ask a basic question. Is the emission flat or does it show a pattern? In whole body and facial measurements, what they often report is a pattern change over time rather than a steady line. The signal rises and falls across the day, and the timing of that rise and fall can be compared to other daily rhythms measured in the body. That matters because a rhythmic pattern is a signature of physiology. It suggests the emission is tracking something living and time linked, not random noise. A third move is to map the surface. Instead of only counting photons, researchers use imaging so they can ask where the emission is stronger or weaker. What they often find is that the signal is not uniform. Some regions show consistently higher emission than others, and those hot spots can be stable enough to compare within the same person across days or across conditions. For example, imaging studies commonly report higher emission from the face compared with other regions, and within the face, some areas show higher signal than others, including regions like the mouth and cheeks. Some measurement maps also show relatively higher emission from the hands compared with lower emission areas, which give researchers another practical surface to compare across time or conditions. This suggests that the emission is tied to regional physiology. The body is not glowing evenly, it is expressing a pattern of light. Those three moves, stress response, daily rhythm, spatial mapping, they do not solve the whole story, but they do something important. They allow us to suggest that the signal is likely multi source, and different tissues can contribute differently depending on what is happening in the body at the moment. Research like this has turned this phenomenon from a curiosity into a testable curiosity. Speaking of testing, let's now look at what recent developments have been taking place in the field over the last several years. One of the most interesting directions is brain adjacent recording. In dark conditions with ultrasensitive detectors positioned near the head, researchers report that the optical signal is not just a flat background. It shifts with simple tasks and state changes. And in their analysis, it shows meaningful relationships to brain rhythms, which is exactly what you would hope to see if the signal is tied to physiology. That brain state thread also connects to a smaller set of studies that look at internal states more broadly, especially emotion and meditation. In one emotional provocation experiment, the reported finding is that the signal increases during anger compared with the relaxed baseline, framed as a proof of concept that emotional arousal can leave an optical footprint. In meditation work, the reported change is not necessarily a dramatic glow, but a measurable shift across the session, including changes in the structure of the signal over time, as if the underlying chemistry becomes quieter during and after the practice. Another recent theme is that external light exposure can change what the body emits afterward. When researchers compare different illumination conditions and then measure ultraweak emission, they report the signal can shift depending on the exposure. In parallel, there are also experiments in cell cultures where red and near infrared light are used as a controlled input. The result is a changed emission pattern alongside changed markers of cellular states. This is a very interesting finding indeed. But the direction that surprised me and grabbed me the most is the one that goes after everyone's source, our DNA. There are many potential biophoton contributors that we talked about so far, but one experiment in particular is suggesting that DNA itself can be the source of biophotons. Now, these results occurred in very controlled conditions, so further research would be needed to know whether that is occurring in our cells. This means that DNA might be included one day in the list of things that can produce ultra-weak photons, such as membranes and mitochondria. It hints that some of the signal could be coming from deeper, more central parts of the cell than many people assume. And then, at the far end of this topic, there is the boundary experiment, the boundary between life and death. Imaging studies in animals report a clear contrast between living systems and postmortem conditions, with the emission decreasing after death, and stress-linked changes in tissue that track with the idea of active chemistry versus chemistry that is fading. It is one of the clearest ways to feel what these researchers are claiming, that this faint light is not a metaphor. It is a measurable readout that changes when life changes. Most of the papers I use for this video will be in the description below. If you want to stay up to date on this topic and more, be sure to join the community. And if you think information like this matters, please let the algorithm know. In the next episode, we're going to take this boundary experiment we just talked about and take it into a more mysterious direction and look at a potentially related phenomenon from the human side. In short, I wanted to know if at the point of death if anyone has reported seeing a light, a release of light. So be sure to set your notifications. The answer may surprise you. Until next time, take care.